Welcome once again to Henry's Impact International Academy, and today we are going to be continuing from where we stopped on our microbial culturing lecture. Alright, let's dive right in. So in our previous lecture, we've been able to explore the concept of cultural medias and different types of cultural media. And in today's lecture, we are going to be looking at the types, we're going to be exploring the types of culture that we have. And as well, we're going to be exploring what we call cultural methods. Cultural methods, which JUPEB can also refer to as techniques. You have to take note of these terminologies. Now, what are the types of cultures we're going to be exploring? We're going to be looking at what we call the batch culture. That's what we call the batch culture. After we explain the batch culture, we're going to explore what we call the continuous culture. The continuous culture. Then we're going to look at what we call the pure culture. The pure culture. And of course, having the pure culture means that we're going to have the mixed culture. Don't be tempted to write in pure culture, nothing like that. The mixed culture. So basically, these are the four types of culture that we will be exploring. And then when we come to culture methods, we are going to be looking at three major types of culture methods, or three major culture methods that we have. We are going to be exploring what we call the spread plate method. After talking about the spread plate method, we'll go down to actually explore the streak plate method. The streak plate method. When we're done exploring the streak plate method, then we will now look at the pour plates method. I just need to clarify something. You know, it's a common error that people make whenever they ask you type of culture. Be careful not to start mentioning type of cultural mediums. So very, very important. These are the type of culture. Remember that culture itself is the population of microbes that are present within that media. The media is just the nutrient supply that will serve or aid the growth of these microbial cells. So you have to take note of that. Very, very key, very, very important, very vital. All right, good. Let's dive right in. Now, we'll start from the batch culture. When we hear the word batch culture and continuous culture, they actually have their explanation or their presentation have a common concept of discrepancy that it can serve in being able to distinguish between the both of them. And what's that? In batch culture, you are having a fixed supply. You're having a fixed supply of nutrients. Fixed supply of nutrients to the culture. Good. So to the culture, you are just adding a fixed supply of nutrients. That means if I'm just supplying 50 grams or 5,000 grams of a particular kind of nutrient, it's just once I'm going to supply it at the beginning and put the inoculate the inoculum into the mixture, and that should just be it. So every other thing we'll be doing is observation. And in what sense of observation are we looking at? We're going to be looking at how they grow. Since you're not going to be involving or imputing any extra feed or any extra nutrient into the culture, we'll be observing this kind of growth presentation. And what's that growth presentation? That's what we refer to as growth phase. As growth phase. Very, very key. What are the growth phases that you're going to be seeing? You're going to be seeing the presence of what we call a lag phase. Are you getting me now? After the lag phase, you'll be seeing what we call the log or exponential phase. The log or exponential phase. After which you'll be seeing the stationary phase. The stationary phase. This is very key. Is in the syllabus. That's what you're seeing here. Growth phases. Then after the stationary phase, you will now have the what? You will now have the death phase. The death phase. Good. Now, this is the growth phase for a batch culture. The growth phase for a continuous culture does not present himself like this. So I'm going to put it there. Growth phase for batch culture. Because in the growth phase for a continuous culture, when we get there, we'll find out that it's just an exponential curve. Alright, good. Let's look at this now. What's happening during the lag phase? You've put in a fixed amount of nutrients into the medium. So during the lag phase is where adaptation is occurring. So the microbial cells are adapting. Are we together now? They are adapting to their new controlled environment. They are adapting, trying to feed and all. Now during the log phase, this is where there is what? There is cell division. This is where there is cell division. The cells are dividing, and when they divide, that will be causing a what? An increase in the what? In the cell population. After which you come to a point where we have stationary phase. Now you remember that this cell division and increase, that is cell growth, because when you say cell growth is an increase in the population of the organism. It's not an increase in the size of an individual cell. It's an increase in the population. Now, in this lag phase, what is contributing to the increase in the cell? The increase in the cell is as a result of what? Of utilization. Of the utilization of nutrients. And I want to believe something. As these cells utilize nutrients, as they increase in number, there will be a point where the nutrients available will no longer be sufficient enough to suit the carrying capacity or the population that is now present. And that will lead to a point where you'll be noticing that the death in the population now is now equal to the what? To the rate of growth, or what we call the growth rate. After this phase, what you'll be having is what? Is a decline, which is the death phase. And the death phase as a result of what? Is as a result of increased accumulation, increased accumulation of wastes, products, or toxins. Obviously, just like in humans, when you eat, the next thing you're doing is defecating, and those are waste products. So increased accumulation of waste or toxins, and that's what will lead to death. And you know, as there is increased accumulation of waste, there is decreased, because the nutrient that is being used up, decreased nutrients. That's what explains death phase. So if you want to even present it in a curve, what will you be seeing? You will see something like this. You'll be seeing something like this. You'll be seeing something like this. If this is where we are recording the population size, the population of the organism, and maybe this is against time, you will notice something like this. You started from a lag phase where there is no significant growth to an exponential phase where the cell numbers are increasing to a stationary phase, after which there is a what? Decline, which is the death phase. So if I want to represent or label, I can call this place the what? Lag phase. I can call this place the what? Log phase or exponential phase. Then we have the stationary phase, and I then have the what? The death or phase of decline. But in a continuous culture is one where there is constant supply of food. So in this one here, there is a constant supply. There's a constant supply of nutrients. We don't call it food. Of nutrients. So since there's a constant supply of nutrients, there is also constant removal. There is constant removal of waste, and that keeps the bacterial cell in a constant exponential phase. Now, in pure culture, it's just a culture that contains a single species type of microbe. So you just have a simple one, and the mixed culture contains numerous species, which is found in your natural habitats, and even in blood samples. So in your blood samples, you have different kind of microbes there. So that's for the type of culture you have: batch culture, continuous culture, pure cultures. And your pure cultures they arrive from single cell growth. Then mixed culture, then numerous amount of cells. All right, good. What are the culture methods that we have? We have the spread plate method. So in the spread plate method, I'll just be demonstrating it to you. In the spread plate method, what's really happening is this. You have your petri dish, for instance. Now, I get what I'm saying now, and then you introduce your agar. You introduce your agar solution. Introduce agar. Then when the agar solidifies, on top of the agar, you will inoculate. Inoculate means you are introducing the specimen. The specimen I'm introducing is called the inoculum. So you inoculate the inoculum, which is now the culture of the bacterial cells, right, into the center. In this place now, you're having the agar solution, right? So you just introduce the this thing into the center here. So with the help of a curved rod, now, which is called the spread rod, you then spread. So I'm going to carry this thing out and I'll be spreading them evenly. So the next thing you want to do in the spread plate method is that you now spread the culture now evenly. So you spread the culture evenly so that this guy here, you just spread it round with this glass rod, and so that this thing here can now spread round evenly. When that happens, after a period of time, the result will be something like this. You'll be seeing holes on that's tiny colonies, because each cell, you know, that was the culture. Each bacterial cell will go and start growing its colony, tiny colonies, either now of identical cells. So this thing here is used in producing what we call pure cultures. So the spread plate method usually is useful in producing the pure cultures. How about the streak method? For the streak method here now, in the streak method, you have the what do you call it? The petri dish, and you introduce your agar as usual to the petri dish. So you have your agar solution there in petri dish. What you now do is that to the edge, you introduce a large amount of the bacterial culture. So I've introduced the bacterial culture or the microbial culture. And when you do that, then you will now streak from the edge in constant directions and produce new streak from the previous streak. This is what I mean. Maybe I have a rod, a tiny glass rod. I use the tiny glass rod and do like this. Spread this thing from this region here. And I believe if you are following me, you know that here will be thicker. You get a larger spread here. So from the next place here, you will now carry the rod again, and from this edge, create a new streak. So this is like your streak 1. So you create a new streak. Obviously, since you are producing the next streak from this one you spread here, the streak you get in this place will not be as thick as this one, right? So you'll be having something like this. Let's say you'll be having something like this. You'll be having something like this. It won't be as thick as the other one here. So you produce the streak there. If you want to create this streak 2, if you want to create another streak here, you have something even less thinner. You have something even thinner than that one, because you are producing the next streak from this one here. That's streak 3. It's just like spreading what you have here from here to here. You can't get more spread compared to what you have here. The quantity will be reducing, so the population will be reducing across the streak. Now at this point here, you'll be having very, very minute amounts of bacterial cells. Each cell will start growing to their colony. So this streak 4. So you are producing the next streak from previous streak. That is what happens during the streak plate method. Finally, the pour plate method. In this pour plate method, you are just going to be having your petri dish again, right? Then you have your liquid agar. You heat it up to 45 degrees. I get it now, which some bacteria can survive in. So you have that there. You have your agar here. It later solidifies, and then before it solidifies, you introduce before solidification. You introduce what? You introduce your diluted, it must be diluted culture of bacterial cells. You introduce it into it, either now in drops, and then you will now rotate it in a given direction of your choice, probably clockwise or anti-clockwise. And I have it there, I just rotating it and rotating it. And then when you do that there, you will get, after doing that, you keep it to dry and cool. When it cools down, you will observe the growth of microbes on them. So that's culturing method. So we've looked at the 3 types of culturing method, and I've explained the 4 types of culture we have. So with this, we've come to the end of culturing lectures, and we will be going into reproduction in the different kind of microbial cells. Thank you very much. Stay tuned. Don't forget to subscribe to our channel and hit the notification bell so that we can drop more content like this. With what we are doing right here, you're sure and guaranteed of high performance in your JUPEB examination.